Humans are the most complex creature of the natural evolution. Yet the human brain is the most complex thing that we have. I always tell my colleagues and students that studying brain is a great joy and a privilege, but also a great responsibility, because I believe that brain is the most complex thing that the natural evolution has created so far. But it is also very fragile. That is why it is important to study brain disorders and do it in many ways. The clinicians, the basic scientists work together in what we call translational neuroscience to untangle, resolve the brain puzzles. Can we understand how the brain works? Is it even possible to do so? A Nobel Prize winner once was asked by a journalist, "Professor, can we understand the puzzles of the brain completely?" And he said, "I hope not." So many scientists, many neuroscientists before me, and many neuroscientists after me will continue to untangle the puzzles of the brain. But the way how science works is you generate a piece of knowledge, and then someone else takes over, picks up where you left, and does a better job. So this is how science really works. I will share with you today some of the ideas and examples of what we can do to advance this knowledge. Science is a very demanding lady. The progress of science, as we call it, the train of science, never stops. It moves full speed ahead, full speed ahead. Of course, we can study brain at multiple levels. We can study molecules and genes. We can study cells, circuits. We can study whole organs, organisms. But it all boils down to behavior, because that is the most important thing that the brain produces. Everything that we do is behavior. Complex behavior, and even more complex behavior. Our hero today is a small little fish, zebrafish, or Danio in Latin, Banma Yu in Chinese. The age, 400 million years of evolution. It's a very old animal, very wise. It's a small fish, freshwater fish that lives in Southeast Asia, India, Thailand, Bangladesh, and surrounding countries. The role of this species today, it is number 2, after only mice, the most useful species in biomedicine. In 5 or 10 years, it may become number 1. So I want to share with you some of the insights that this animal can give us. And in fact, the structure of zebrafish brain is not that different from that of mammals, mice, rats, monkeys, and humans. And they do have a lot of neurons, about 10 million neurons. All the chemical important molecules are there that humans have. In fact, we share about 70% of genes with zebrafish. So we are 70% zebrafish. But if you look at the disease-related genes, all the genes that are associated with human diseases, the percentage is even higher. It's about 90%. And if you look at the conserved meaningful sequences in each gene, the level of similarity is even higher. It's almost 98%. So this fish is very similar genetically and physiologically to us, and therefore we can use it to solve some of the brain problems. Zebrafish are brave because they respond to stress the same way as humans do. When they are afraid, they dive, they freeze, or they begin to move erratically, trying to raise the cloud of debris from the bottom and hide from the predators. Is it not so reasonable and logical and beautiful and smart? And even the stress hormones that we humans use are the same. So, for example, our main stress hormone is cortisol. And zebrafish use cortisol as well. And when they are stressed, their cortisol levels are elevated. When they are calmed down, like in humans, their cortisol levels go down. What is even more interesting, the time course, how the cortisol levels rise and go down, is almost identical to that of humans. A scared fish and a scared human will rise cortisol in 15 minutes, and then if the danger is gone, the cortisol levels will gradually dissipate over the period of an hour and a half. This is how we humans respond to stress, and this is what zebrafish do as well. So, can we use this model to understand some of the brain puzzles? Let's think about stress. We can model stress in zebrafish and learn some lessons about it. So, for example, if you apply some very unpleasant stressors to zebrafish for 90 minutes, the zebrafish will remember that for at least 10 days. When you analyze their behavior, they will be, of course, anxious 10 days because they remember this stress. But let's see what happens at the molecular level. If you look at the expression of important genes, you can say that the genes that are markers of microglia and astrocytes are upregulated. Now, what is microglia? What are the astrocytes? These are additional cells in the brain, not neurons. The glia that helps the neurons to work properly. So these findings show us that stress changes not only neurons, as we would expect, but it also changes microglia and astrocytes. The other interesting finding was that there are some genes that are markers of neuroinflammation that were upregulated in zebrafish exposed to such stress. Meaning that stress is not about the chemistry in the brain, it's also about the inflammation. So the brain needs to respond, and stress causes the inflammation of the brain. Another interesting finding was that stress also increases what we scientists call apoptosis, which is the word for cell death. So stress actually kills some of the zebrafish brain cells, and we see that by elevation of the apoptosis biomarkers in the brain. So these lessons tell us that stress has a very complex effect on us. And because zebrafish are similar to humans, we can translate that knowledge to our clinical situation. So stress is not only about the chemistry of the brain, it's also about inflammation, it's also about neuronal death, and many other things. But the most important part of that is we can use zebrafish as time machine. So if you can identify molecular pathways that are shared across humans, mice, and zebrafish, that means these pathways are the most evolutionarily conserved, complex, that are involved in a brain disorder. So then the next generation of the drugs can be developed based on targeting these old, evolutionarily conserved molecular machinery. I think this is very important, especially today when the innovation is, of course, blossoming, but yet we have all the drugs that we use right now that are 30, 40, 50, and some of them 70 years old. We need new drugs that would work on totally new mechanisms, and zebrafish can offer the window of opportunity for such research. The problem is that we don't have good drugs. We do have many candidates. We just don't know which candidates to use. So the chemical laboratories come up with many compounds that can be life-saving drugs, yet we don't know which drugs will be actually working. So you need a biological system to test it. If you do this in mice and rats, that will take years. It will also be extremely expensive. Zebrafish screens have the capacity of high-throughput drug screening because they are small, space and time efficient, and much cheaper. You can speed up the drug screening by testing hundreds and hundreds of compounds per day. So we can eventually find some promising candidates. But even if this is done, this will not be enough because there will be more and more compounds discovered every year. So the question is, can we have some help, perhaps from artificial intelligence? The idea is very simple. I'm a neuroscientist. I will talk about the brain-altering drugs today. But the same principle can apply to other drugs treating cancers and infections and many other diseases. We can analyze drug responses in zebrafish, create a library of these responses, put all this library, all this data into powerful computers. The computers will use some deep learning protocols and come up with the solution. This is the general theory, but the devil is in details, and it's easier said than done. So we tried to work in this direction, and there were several important steps that we needed to take before a success could be possible. The first step was to create the real three-dimensional representation of zebrafish drug responses. Just understand that humans and rodents, we live in a two-dimensional space. We walk in 2 dimensions. We don't use the third vertical dimension very much, but fish do, birds do. So the real movement of the fish is much richer than ours. It's actually 3-dimensional locomotion. Now, we can give different drugs and understand what typical response these drugs will produce. Here are examples of what different drugs will do. And as you could see, different chemistry, different pharmacology of the drug will produce different responses in zebrafish. These responses will be consistent. The same drug will do the same thing, more or less, in this zebrafish, and that zebrafish, and this zebrafish, and that zebrafish. So these responses will be consistent. We accumulated hundreds of responses from dozens of compounds, tested all major brain-altering compounds in my lab, and then tried to do the next step. We applied artificial intelligence to actually analyze all that variety of responses and try to cluster these responses based on the effects. And this is the example of what we got. And in fact, that was a partial success because some of the drugs that had similar pharmacology were recognized by the artificial intelligence as similar drugs, which is a good news, so that similar acting drugs were correctly recognized by the artificial intelligence in zebrafish as similar. So that was the very first application of artificial intelligence for discovering of psychotropic drugs in zebrafish. And then more recently, another important step was made when we created a very sophisticated artificial intelligence platform for screening drugs that looks very complex, but essentially this is a neural network that has two parts, the head and the backbone. So the backbone is where the analysis occurs, the learning occurs, and the head is where the prediction of the drug classes occurs. We did a very simple experiment. We took 4 drugs, all very relevant to us: caffeine, nicotine, alcohol, and water for the control fish. Just only these 4 drugs. Well, we knew these drugs will work very well in zebrafish. We tested them multiple times. But the question is not what we know. The question is, what does the artificial intelligence will know? So we collected data from about 16 fish and then fed this information to the artificial intelligence. You could see at the bottom the learning curves as we cycled the data again and again and again 200 times. With each time, the learning was better and better and better till it reached a plateau. So the artificial intelligence really learned something. But the question is, will it recognize the drugs when we give these drugs to zebrafish again? So we had a new group of zebrafish given the same drugs. Now it was the time for exam. Now it was the time to test whether our system could actually correctly detect the drugs we gave. And the results were astonishing even for us. With the precision of 100% for most of the drugs, except for alcohol, the artificial intelligence was able to predict absolutely correctly the drugs that we have given. What does that mean? That means that this method can be applied to screening any compound. And in fact, we made this system available publicly to anyone to use. We uploaded the architecture of the network. We uploaded the codes in a public domain, so any lab in the world can use it for their research purposes. Of course, it's the proof of concept study. The real science will be more difficult. More drugs may cause lesser accuracy of prediction, but the rule is, it's like with student learning. The more knowledge you put in the brain, the better the brain can understand the concept. Same applies to the artificial intelligence. The more data we feed to the artificial intelligence, the better and more accurate the prediction will be. But the real science doesn't work like a German train. The real science is more difficult. You may have many, many interpretations for the same phenomenon. So the journey begins. The train of science doesn't wait for anyone. The train of science moves full speed ahead. Thank you very much.